Swine It.
It's time for a new era of communication in the swine industry, one that you can get the latest updates while commuting or driving to farms. Here you will have the brightest minds of the global swine industry in your pocket.
No one really cared a whole lot to fund African swine fever research when it was in Africa. And now that in countries that are major pig producers, it's a big concern for everybody.
Swine It podcast is only possible with the support of forward-looking and innovative sponsors like Elanco's Prevacent, a new PRRS Spective. Visit Prevacent, PRRS.US to learn more. NutriQuest, experts serving producers and delivering breakthrough solutions. Genesis, the first power in genetics. Zinpro, essential trace minerals, exceptional performance. Every Pig, a simple yet powerful pig health and production management tool. Gestal, always one step ahead in swine feeding.
Welcome to Swine It podcast. My name is Marcio Gonçalves, your host for today's episode.
This episode's sponsor highlight is about Genesis. Genesis is the largest independent producer of high health registered purebred swine in the globe, having over 80% of all registered purebred breeding stock in Canada. The Genesis genetic program uses genomic selection strategies focused on productivity, faster growth, efficiency, high yield, and meat quality. To know more, go to genesis.com. GENESUS.com.
Hello everyone. Today our guests are Dr. Douglas Gladue and Dr. Manuel Borca, both from the USDA, and they will share the latest development with African swine fever vaccines. How are you, Douglas and Manuel?
Good. Thank you.
Yeah, I appreciate you guys taking the time today to chat with us and our audience about this important subject. So the first question we always have is if you can share, each one of you can share, about yourself, your career so far, and how you got involved with swine research.
Well, I'm the older one, so I can start. I have been working in USDA first as a postdoc, then as an invited scientist, and now since 2001 I'm in a permanent position. And I have been working with the main three viruses that are working here in Plum Island, that is African Swine Fever, Classical Swine Fever, and Foot and Mouth Disease virus. And I have been particularly working with CSF, Classical Swine Fever, and ASF, African Swine Fever, in the last 18 years. Basically, that's it.
Very nice.
For me, I got my PhD on Long Island, and when I was looking for a postdoc, Plum Island was nearby, and I started at Plum Island about 12 years ago, and this became a permanent position about 5 years ago, and been looking a lot at African swine fever and classical swine fever, and looking at virus-host interactions, and that's how we ended up in the direction of looking at rational development for vaccines, and how we ended up with our vaccine candidate.
Very nice. Very, very good. Thanks for that. And so as we jump right into the topic here, for the last several years, as we speak about ASF, what has been the challenge in getting an effective vaccine?
I think the problem with African swine fever is that there have not been many laboratories in the world, around the world, working on African swine fever. Since the disease was started in Georgia, Republic of Georgia in 2007, and first to Russia, and then to Eastern and Central Europe, and now more lately to China and Southeast Asia. I think that there are more and more people paying attention to this disease. And I think the research that had been done in African swine fever back in the 1990s and with the development of recombinant viruses and the possibility of investigating the role of specific genes in the production of disease, open a window in understanding why or how the virus causes the disease and how we can manipulate that in order to produce vaccines, viruses that are attenuated, but they do not produce disease anymore. And that was the key in order to start the work that we have been doing on Plum Island since then, that is the development of African swine fever vaccines that are live attenuated. And the reason probably is, besides that there were not too many people working on this, is that this virus is large, it's very complicated, and we do not know a lot of things that are important to be known in order to produce a vaccine, like what are the host immune response that are effective in eliminating or preventing the disease, or which part of the viruses are important in order to produce immunity.
I think that sums up pretty good. I think the big thing that I always tell people is that no one really cared a whole lot to fund African swine fever research when it was in Africa. Mm-hmm. And now that Africa and it's in countries that are major pig producers, it's a big concern for everybody. So it's getting a lot more attention. Even if you look at the number of papers that have been published on African swine fever versus human diseases or other animal diseases, there's a huge gap in knowledge. And this gap is critical to learn, to understand, to be able to make effective vaccines and to be able to control disease. Right now, we don't know what proteins specifically are needed to have an immune response, and if you don't know that, you can't make a subunit vaccine. So we're kind of in the early stages of understanding this virus, even though it has been around for many years.
Very interesting. So I think you're getting some very interesting research there right now. So is there a light at the end of the tunnel? What are the latest updates you guys can give us on effective vaccine? We
can really only talk about published material. Whenever you license a product, what's out there in the public domain is free game to talk about. We can tell you that we have several live attenuated vaccine platforms that we've made over the past 10 years or so. And these platforms are still in the experimental stage, where they've been tested in laboratory conditions under defined conditions with a small set of animals. They still need a lot of other testing to be able to be approved, to be able to be used in whatever country we're targeting. They're at the very early stages of vaccine development. But with our most recent candidate, and probably how you've came across us, is with 𝐼177L, we're seeing a new gene that hadn't been discovered before, and this vaccine platform appears to have several advantages, where it's offering low dose protection and there's no residual virulence. So if you give a real high dose, you don't see any symptoms in the animal, including any fever. And also we seem to see apparent sterile immunity. So what that means is a vaccinated animal doesn't appear to replicate wild-type virus, and that's something that is pretty good for a vaccine profile for safety-wise, because there's less of a chance for the vaccine virus to ever recombine with wild-type virus. Another thing that we see is that we don't see vaccine virus shedding. So what that means is if we put an animal that is not vaccinated in with our vaccinated group of animals, we don't see the vaccine spread to that unvaccinated animal. So the disease remains in the animals that were intentionally inoculated. So this is a profile we really like for a live attenuated vaccine, but again, it's in the early stages, and it still needs to undergo larger testing and regulatory hurdles before we can proceed further.
Very interesting, yeah, and very exciting. For us that don't really understand the whole process of developing a vaccine, could you step back a little bit and just generally speaking, what are those processes? what's an average timeline or the range? It doesn't have to be ASF specific, but just in general, just so we can wrap our heads around it.
That's not really an easy question to answer. For African swine fever, people historically have made live attenuate vaccines by deleting specific genes in specific isolates, but there's a lot of problems to doing it. It's not an easy task to just delete a gene in a genome of ASF. And the reason why it's not easy, or one of the reasons it's not easy, is that so far African swine fever can only grow in primary swine macrophages. So these cells have to be constantly isolated from a donor animal, and they have to be used within a week.
Wow.
So it's not like other diseases where you have a stable cell line and you pass cells, you can do all the experiments you want. You're limited to the number of macrophages you have, and you're limited to the time frame for those macrophages. It's also making a recombinant live attenuate vaccine is to purify recombinant virus from wild type, and this is also not an easy task because there's no quick way to do it. Things can take a long time. Sometimes you get lucky, but it can take over a year from concept to completion to delete a gene with a lot of work every week. So it's not an easy task to make a live attenuated vaccine. So that's kind of where we are. The other aspect is that African swine fever has at least 150 different genes, and probably only about 10 of those have ever been really truly characterized for gene function. So the majority of the virus and the function of the genes and what happens during virus infection and pathogenesis is largely unknown.
Wow. Yeah, that's very interesting. So, and you and I chatted briefly before the recording here that the overall timeline and timeline to be able to produce in large scale as well as cost is not something that you guys would have an idea today, right?
No, that's a complex question, and we get asked it a lot, and it's impossible for us to answer because commercializing a vaccine involves a commercial partner. It involves regulatory authorities. It varies from country to country. So timeline is just out in the open. When things get completed, they get completed.
Very good. Well, anything else on the ASF vaccine topic that you'd like to share with our audience? There's a lot of veterinarians, nutritionists, production folks from around the globe really in the audience. Anything else you would like to share?
No, I would say that probably just remark of some of the things that Doug said in terms of the difficulty of producing this kind of vaccines, and maybe just to give you an idea, when you go through the literature that is published regarding the vaccine candidate that may produce protection against the acting strain of virus that is producing the epizootic in Eurasia right now, there is no more than 4 or 5 papers that show that could be a candidate vaccine. And if you go a little bit in depth and in the analysis of each of these 4 or 5, maybe 6 at most papers, you see that most of the virus has some problems in terms like no complete level of attenuation or the need of using high doses of virus to produce protection. So that's why when Doug was talking about this last candidate that we produced, the 𝐼177L, that has those characteristics that he described, that's why we are quite excited about that one. Because apparently work at very low doses and is very immunogenic, and the level of protection that is induced is very high. I think that there's not a lot of things out there right now in terms of potential vaccine candidates that protect against the virus that is producing the problems right now. One thing that we should probably mention is that immunity against one virus does not cross-protect against other African swine fever isolate, and that is a very important issue. So every time or almost every time that you want to produce a vaccine against one particular isolate, a strain of virus, you need to produce a vaccine using that one. It's a big problem because you almost have to produce a vaccine for each of the isolates that is out there. We do not understand so far what are the basis of the lack of crop protection among different isolates at this point.
Okay. And so I guess one question I have is, it sounds like it was very interesting findings, and based on your experience and your level of confidence on this one gene that you guys described, just for my simple mind, is what could go wrong still?
There are a lot of regulatory experiment assessment that had to be done with the vaccine. With a repeating dartboard, what we have is a very good result at the laboratory level, in an experimental system, and from there to have something that we say this is a very good, stable vaccine. We had to go through several experiments that will show, for example, that there is a genetic stability in the mutations that we introduced, that the virus does not mutate further, that there is no reversion to virulence, that the virus will not recombine with any wild-type virus out there. So there are things that need to be tested, and again, we would probably say that we are in the very beginning of the story right now.
The truth is, precision swine production is not the future, it is the present. Every Pig is the intelligent pig health platform. It is a simple yet powerful pig health and production management tool. Request a free 20-minute demonstration at www.everypig.co/swineit.
It is time to our famous three. Well, so then at the end here, one thing that I would like to ask all the guests that we have in the podcast is, one is, what's you guys' favorite pig-related book or resource?
Lately, I've been looking at Pig Progress. That's been a pretty good resource, in part because they seem to be the most up-to-date for African swine fever outbreaks, where they're reporting pretty quickly, and they keep maps of all the outbreaks publicly available. So they've been a pretty good resource, at least for me.
I don't have any, actually.
I'm sure lots of scientific papers there, Dr. Borca.
Well,
very cool. How about you guys' favorite book in general, outside of pig production?
I don't read a lot of books. I'm more focused on the scientific literature and keeping up to date with what's going on in the world for the diseases that we study. So I don't have a book in mind right now.
And then what is actually a very difficult thing to do, because there are many things coming every day. So most of the time you have to try to be updated on that, and it takes most of the time.
That makes sense. And then the last question we have is, what, in your guys' opinion, what do you think sets apart successful swine researchers or professionals from those who are not?
I think in our field of research, there isn't really a real gauge of success or non-success, at least for people doing stuff like we're doing, at looking at different host virus interactions or testing potential vaccines. I think that there's a lot of luck factor, and to say someone's successful or not is hard because there's no real measurable way, at least in my opinion, for that. I think for everyone, you get what you put into it a little bit, and hopefully you get out what you think you should be getting out for the work you're doing. But at least in our field, there's not a whole lot known, so it's an open book for learning new things about the virus.
Interesting, yeah. That makes sense. Any thoughts there, Dr. Borca?
I will basically subscribe what Doug said. It's a lot of, especially working in African Swine Fever, there are so many unknown questions that sometimes you have to be a little bit lucky, and You're trying to be systematic in the way that you work, but this is a very complicated virus. It's a very difficult virus to work with. So frustration sometimes is very frequent.
That's awesome. Yeah, I definitely agree. Great. Appreciate you guys' time. I know you're very busy this time, with there's a lot of things going on in ASF world. So appreciate, and I have no doubts everyone's going to be rooting for you guys there on the finishing developing this vaccine.
Okay. Thank you.
All right. Thank you.
Hey everyone, we just crossed 15,000 downloads of our episodes, and I wanted to say thank you. Please share our episodes with as many people as you can so we can continue to impact the life of swine professionals from around the globe with the wisdom of our great guests. Before you go, make sure to get in our waitlist for the Swine Talks Web Conference, the first online conference of the global swine industry, an update on hot topics, and we even gonna have some controversial topics of the global swine industry, so you can leverage that knowledge in your day-to-day. Go to swinetalks.com and get on our wait list. We'll talk soon.